Dear ladies and gentlemen, the talk Ecotoxicology of Microplastics, Knowledge Gaps and Possible Steps Forward is presented by Katrin van Pothoff and myself. My name is Christian Laforge and I am, among other things, the spokesperson for the interdisciplinary project PLAVIS. In PLAVIS, we investigate and model the pollution with microplastics across ecosystems in a large European river basin, the River Weser National Park Wardensee Model System. In particular, we study how much plastics is introduced into rivers via terrestrial ecosystems and how much is then transferred into the marine system. We further study the effects of microplastics on both aquatic invertebrates and the interaction of pathogens with microplastics. The results will be used to develop novel teaching materials to provide an education platform for teachers, pupils, and parents across Europe. PLAVIS is embedded in a research initiative of the German Federal Ministry for Education and Research called Plastics in the Environment, Sources, Sinks, Solutions, which started in 2017. A total of 20 joint projects are funded with around 35 million euros and are arranged along 5 thematic areas covering the entire life cycle of plastics. The spectrum of topics covered by this initiative ranges from green economy, consumption, recycling, limbic systems to seas and oceans. Further, so-called cross-cutting issues were established to identify and use synergies among those projects, and Katrin and I are responsible for CCI2 evaluation methods of possible effects of plastics in the environment. Especially for microplastics, there's still a debate going on if it is hazardous or not. And this is nicely depicted in the table of Bucci et al. Showing that even the same polymer type tested in the same science range resulted in negative effects on some organisms in some studies and in some not. One aspect to consider when interpreting the results is that some studies have used very high microplastics concentrations. Another important factor is that many studies have used pristine microplastics that has little in common with the microplastics found in the environment. However, when assessing the risk of microplastics, the most important factor is that microplastics is not microplastics. It consists of different polymer types occurring in different sizes, having different shapes, surface characteristics, and so on, and are also including different additives. And this complexity must be included in ecotoxicological testing. And now you will hear more about the recommendations of the CCI in the forthcoming slides from Katrin.
Hi, I'm Katrin. I'm coordinating the project Microplastics, which is about microplastics in dams and reservoirs, its distribution and effects. In addition to the inherent heterogeneity of microplastic materials mentioned before, microplastic in the natural environments is not naked, but associated with microbes. Our focus is how this influences the particles themselves, their aggregation and sinking, and their food quality. Moreover, we study how these particles influence the microfauna, myofauna, and macrozoobenthos, both as single species and as parts of the freshwater food webs. Key organisms we study are rotifers, nematodes, and snails, which you see, but please note the images are not scale adjusted. Within the cross-cutting topic 2, the authors of this presentation attempt to bring together topic-related results from the joint research projects of the research focus and other projects which are similar. We want to discuss and coordinate possible evaluation methods and coordinate common positions and products to inform the public about the effects of plastics in the environment. One important recommendation is the use of non-plastic reference particles in microplastic toxicity tests in order to account for general particle effects. You see here results of a feeding experiment with nematodes. The endpoint was inhibition of reproduction, and polystyrene and silica beads of different sizes were applied in combinations with food bacteria. In the graph, you see the lower the ratio with an effect is, meaning the more left side the curve is, the stronger was the inhibitory action of these beads. So the strongest inhibitory effect occurred with 10 micrometer polystyrene particles that cannot even be taken up by the nematodes because their mouth is too small. Silica particles have lower effects than polystyrene, especially with area-based evaluation, which you see in the right graph. This is maybe because of their higher specific density. The most likely explanation for these findings is that the beads reduce the availability of food. This is one of the 3 possible mechanisms of microplastic action that have been substantiated so far in a recent critical review. The other 2 were internal and external physical damage. In order to confirm other suggested effects, future studies should follow a list of minimum reporting criteria, which we compiled within the cross-cutting topic 2. What we can learn and conclude is that mechanistic understanding of particle effects is still insufficient. To overcome this, it's necessary that all available information about the particles used is reported. Moreover, it's necessary to use non-plastic particles to control for general effects of solids. And the publication of well-documented no-effect results is highly recommended to authors, editors, and publishers. With this, I would like to thank you for your attention, and Christian and I will be happy to take your questions.